Are people in like a waiting room? I don't see anybody
Yep.
coming on.
Yep.
Okay, they'll come on in a second.
Yep, they're coming.
Ah, there they are. I see. Good. Lots of people coming in. Good. For all those joining, we're going to give a couple minutes and then we'll get things started. For those joining us, we're going to start in just 1 minute. And a few more people get a chance to get on. All right, we're going to get started. Welcome, everybody. This is our second in our series of webinars about nitrosamines. For those who don't know me, I'm Ken Waterman. I say the word Chuck because so many people do know me. I'm the president of FreeThink, and I'm very happy to be helping to facilitate this webinar series with an outstanding set of speakers. I'll be introducing today's speakers, but as you can see here, our series, we had already Maria Ultramari present on the questions of toxicity. Today I'll be introducing in a minute Rachel Hemingway about in silico tools. We'll then have in a couple weeks Gary Scrivens, more about the chemistry, the rates in solid state. And then finally, Steve Baertschi on really what's the chemistry behind it all. So these webinars are all recorded, and anybody interested in getting access to the recordings simply can do so through our FreeThink website or sending an email to us about it. Also, the PDFs of each of them are available. The recordings do have closed captioning, so if you have a little trouble with the English, you'll be able to read it as well. Before introducing our speaker, just maybe a quick kind of advertisement. So FreeThink's your sponsors. So we like to say we use world-class science to speed better products to market. And we have a number of offerings. We have our ASAP Prime software, which if you're not used to is very powerful tool for accelerating shelf life determinations, including of N-nitrosine formation, and our laboratories as well in the United States. We do also develop drug delivery technology, and we've just introduced a first oral osmotic controlled release capsule. So anybody interested in that, as well as other technologies. Before I introduce Dr. Hemingway, Rachel, I want to mention that, this issue of nitrosamines has caught a lot of us very quickly as a major factor in our pharmaceutical development. And this has only been a few years. Whether this is an over-exaggeration of a problem or not sort of becomes irrelevant to us because the health authorities in the countries are now limiting it. So a lot of what we're talking about is how to know whether there's a problem, how bad is the problem, what am I going to do about it will be another part of this if there is some issue. So with regard to that, today's speaker is Dr. Hemingway, and I'm only going to give you a snapshot of who she is. I've known her, Rachel, for a number of years, and she graduated from the University of Hull with a PhD in chemistry, developing tumor-targeted therapeutics. She then worked for a couple of years at a chemical manufacturer before she came to Lhasa, where she's been now for more than 8 years. She is one of the key drivers of the program called Zenith, and she'll be talking about that, so I'm not going to go much into that, but in general, this is software allowing people to know what might happen from their drug. So what kinds of things are occurring, what kind of chemical reactions, and they do a good job of constantly improving it. So I think over time, we'll see a lot of that. She has co-authored 5 articles, 1 more in preparation, and 4 book chapters. She's going to be talking to us today about how to use in silico methods. Before I turn it over to Rachel, I want to let everyone know everybody is automatically muted on this webinar. If you do have questions, and I do encourage you to ask questions, please type them into the Q&A box. At the end of the webinar, at the end of her presentation, I will judiciously choose which ones to read off and ask Rachel to answer them. And then any questions that didn't get answered, we'll go ahead and try to get answers to people outside of the webinar as well. So please, you don't have to wait till the end to ask your questions. As you think of questions, you can just go ahead and type them in. If they're referring to particular slides, you can go by the slide number. And with that, I'm going to turn over everything to Rachel Hemingway, and she will go ahead and do her presentation.
Thanks, Ken. I'll just get my slide sharing. Okay. So I guess just I would like to start off by saying thank you to Ken and FreeThink for providing this platform for us to talk about nitrosamines. I'm very humbled to be part of this series alongside some fantastic speakers. So I hope to provide an informative talk today, kind of building on some of the topics that Maria discussed in her webinar a couple of weeks ago. But I'm going to focus much more on sort of the chemistry side of things, whether a nitrosamine can form, and if it can, what do we do about it? So I am going to go off video for the duration of the webinar, just because I'd like the focus to be on the slides and definitely not my face. And then I'll come back on for the Q&A session at the end. So I'll just get my laser pointer. Push on my slides. There we go. Before I launch into the body of the talk, I'd just like to highlight who LASA Limited are, for those of you in the audience who haven't heard of us. LASA is a not-for-profit organisation. We are driven by our purpose, which is to enable informed decision-making on chemical safety. In line with this, our objective is to create software technology which streamlines compound development and minimizes animal testing. Our technology is designed by scientists for scientists, and that's done in collaboration with industry stakeholders and regulators. And so what we do in a nutshell is we create software. We create software solutions with the aim of supporting decision-making around drug safety and quality. And the logos that are outlined are the sort of the suite of software solutions that we develop, and you can find a full explanation of those on the website if you so wish. Before I launch into the body of the talk, and I guess just to ensure that everyone is in the right room, I'd like to highlight some of the keywords and the topics that are associated with today's webinar. So I will be covering the use of a predictive in silico tool with respect to nitrosamine degradant formation. I'll also be discussing nitrosamine drug substance related impurities, so-called NDSRIs, and I'll highlight how a database of nitrite levels in excipients can be utilised to help understand and potentially mitigate nitrosamine issues. So I pinched the title of this slide from the title of the paper that the fig came from. It was published in OPRD a few months ago, authored by Raphael Nudelman et al. And I've seen this kind of timeline in quite a few publications, but felt this was one of the more up-to-date versions, if you can be up-to-date in the nitrosamine world. And so where the nitrosamine saga began was with NDMA detection in valsartan back in 2018. This led to a cascade of events really around the detection of so-called small dialkyl nitrosamines. And NDMA was found in several other marketed drugs, which led to their recalls and sometimes suspensions. This happened around the sort of 2020 mark. Guidance updates followed quite swiftly after. And then moving into 2021, another key event that changed the landscape even further was the detection of NDSRIs that were found in several drug products. Mostly coordinated responses from the regulators, and updates on guidelines followed from the EMA, the FDA, Health Canada, and the MHRA, with the latest publication by the EMA on acceptable intakes for some NDSRIs. That was sort of August time, but updates keep coming through. And Maria elegantly went through the CPCA rules for acceptable intakes in her webinar, so I won't really be touching on that kind of any further. But all of that makes for quite a problematic and a challenging picture for all industries associated with drug discovery and the drug development pipeline, as well as regulators. So what poses the challenges associated with nitrosamine formation? Well, I think it's been summarised quite nicely in this figure from another OPRD publication. And there are 3 risk factors, and all are required. Firstly, there needs to be a secondary amine source. And I guess I should say it's usually a secondary amine, but actually other nitrogen-containing functional groups can also pose a risk. Then what these authors term conducive conditions, and this is a term that I'll continue to use throughout my slides, things like pH, temperature, water, and finally a nitrosating agent. These can be found in reagents in the form of nitrites or nitrates, in excipients as inorganic nitrites, or through a degradation pathway of the API itself. So for example, a nitro group, for example. Where can this happen? Well, in quite a lot of places. It can be in the synthetic route of the API, the drug manufacturing process, via a degradation pathway of the drug substance or product, or via an interaction between a drug substance and an impurity, and also from primary packaging materials. There's even evidence that NOx gases in the air can be responsible for nitrosamine formation during specific manufacturing processes, such as drying. So that is a lot of places where an assessment is required to ensure that any nitrosamine risk is either negligible or tolerable. So who can help in this space? Well, there are tools and databases being created to support nitrosamine risk assessment, as well as all the experimental work that's been going on within industry, which can be used to derive models and descriptors, something that Gary will be going into much more detail in his webinar. But Zenith is one such tool that can help support nitrosamine risk assessment. Zenith can be used to predict what's theoretically possible for a given chemical structure based on known degradation pathways. And this provides an overview of that theoretical space for that particular compound. Armed with that knowledge, because as we all know, knowledge is power, that theoretical space can then support and aid decision-making for the potential space for developing force degradation studies, because you can think, well, I already know what I'm sort of looking for. I know where to target my attention. And in turn, having a good overview of that potential space can ultimately aid understanding of the likely and actual spaces with respect to degradation products that you might find in the final packaging and storage conditions, because hopefully nothing needs to be a surprise. So Zenith is a knowledge-based system, and its knowledge base can really be thought of as a library. And that analogy works quite well in terms of it is full of books, except our books are chemical degradation rules. Zenith then uses this knowledge base, this library of chemical degradation rules to match to a relevant fragment in a compound. If there's a match, then a likelihood score is calculated. So how likely that particular pathway might be to have that pathway might happen. And then the output of the system is a theoretical prediction of the degradation that's possible for that chemical structure. The methodology behind the tool is that as a user, you input your query structure, your API of interest, and you define a likelihood level that you're interested in, so a threshold. And it might be that you want Zenith to tell you what's likely to happen, so you set your threshold high. Or you might be at an early stage in your development process, and you want to know everything about your potential lead compound. So you'd say to Zenith, tell me everything you know, and you would set your threshold low. You would then define reaction conditions, and the types of condition that are available in Zenith really mirror those recommended in the ICH guidelines and are typical of those employed in force degradation studies. By typical, I mean in a general sense, so the presence of oxygen, water, light, for example. This information is then used by what we call our transformation engine, which sort of cycles through all of the books in its library and finds something that matches, a fragment that matches. Pattern matching in Zenith works purely on a structural level, the matching of functional groups or fragments. If a fragment matches, if a fragment matches something that's outlined in a rule, in a pattern, then that's considered a match. Each pattern match will then undergo a score calculation to derive a likelihood for that degradant, and product degradants are generated. To put that into a little bit more context, just got very much a representative example of an output that you could expect from Zenith. I chose bromazepam, which has nothing to do with nitrosamines, purely for an illustrative example. So Zenith results are presented in a tree, we call it a tree, which allows for successive generations of degradants to be displayed. So first generation degradants can degrade further into second generation degradants. And this is just a section of the prediction for bromazepam, just purely for illustrative purposes. I've mentioned that predicted degradants also get a likelihood score. So we try to calculate the likelihood of a given pathway, and we do that using a score scale of 0 to 1,000. So 0 would be impossible and 1,000 would be certain. We don't predict 0 or 1,000, we just use those as our boundaries. And likelihood scores are calculated based on variables such as reactivity, the influence of reaction conditions, as well as physicochemical properties. So we have BDE and pK calculators embedded into the software. So I don't have time to sort of explain and go through the derivation of likelihood scores and how those calculations come about. That really isn't the focus of today's webinar anyway. But if you would like more detail, then please do get in touch. So knowing how the tool works is one thing, but what knowledge does this tool have? What is the brain of Zenith? What does that chemistry look like? So the knowledge base in Zenith is about 15 years old now. It's reasonably mature, and to date it contains 635 rules. We classify that knowledge into 6 categories. Oxidations and hydrolyses make up about half the knowledge base, condensations and additions around 20%, and the final quarter is split between photochemical reactions, isomerizations, and eliminations. How are these rules developed? Well, mainly from public literature and degradation textbooks that we review and we extract information from, but also through data sharing initiatives where actual experimental data, which is donated by various companies, is utilised. We obviously anonymise it to protect everyone's IP. How good is this knowledge base? So it might be mature and it might be 15 years old, but how good is it? And I guess I need to define what I mean by good here. Well, what I mean is, how well does that knowledge base predict pathways that are seen in actual post-degradation studies in industry? How well does it cover that space? So in 2014, we conducted a validation study on the knowledge base, which at that time contained 277 rules. And the data set that we used was an experimentally derived data set of degradation products from forced degradation studies that were donated by pharmaceutical companies. Zenith had never seen this data before, and so we cross-referenced those experimental degradants against what Zenith could predict at that time, and we were getting just over half right. A few years ago, we then did a very similar study in 2020, an update, if you like, using a brand new data set donated by pharmaceutical companies, many of the same pharmaceutical companies, I should say. And the knowledge base in 2020 contained 577 rules, so it was almost twice as big as what it was in 2014. And Zenith was able to predict 70% of those experimentally observed degradation products. And so that increase in sensitivity is down to the growth of the knowledge base in a relevant way, increasing its coverage of the degradation field. So onto the meaty part. What about nitrosamines? How are nitrosamines covered in this knowledge base? Well, we've got 16 rules that cover various types of nitrosamine degradation reactions. 15 of these are classed as condensations and additions, and there's also an oxidative pathway. If I start with NN bond formation, the types of chemical class that are covered, and if we look at the green part of the circle first, so classical secondary amines, hydroxylamine and hydroxylamine ethers, hydrazines and hydrozides, and tertiary amines bearing an electron-rich aromatic group would all be predicted with conducive conditions and a nitrosating agent present. These functional groups have a pretty appreciable nucleophilic character, in particular secondary amines. So, yeah, as I say, when conducive conditions are present, then these would be predicted at a reasonably high likelihood on our scoring scale. I guess just to say on this slide, I haven't put all of the definitions of all of the different R groups, because that would have made for quite a chaotic slide. But each of the R groups for each chemical class will have a specific definition. For example, if I just pick secondary amines, R and R1 would be defined as an aromatic carbon or an aliphatic carbon not attached to an electron withdrawing group. And similarly, the other chemical classes would have definitions for their R groups too. So it's not that everything would fire. There is a scope to each of the rules. Looking at the compound classes in blue, so pyrrole type compounds, guanidines and amidines, amides, carbamates, ureas, sulfamides and sulfonamides, and tertiary amines bearing a proton on the alpha carbon. They are all covered by rules in the knowledge base as well, but these being weaker nucleophiles, they would generate a lower likelihood score when conducive conditions are present. In addition to NN bond formation, the knowledge base also has a rule to cover NO bond formation, whereby hydrazines are oxidized in basic conditions to a nitrosamine. And I guess there's a reason why I've put it in blue, not strictly nitrosamine formation, but NC bond formation, namely of enols, enamines, and amidines, are also covered in the knowledge base. These are naturally of much less concern since they don't actually generate a nitrosamine, but I guess I put this down because it can be just as useful to understand what isn't as likely to form as what is. And there is some evidence in the literature to show that sometimes there will actually be a preference for C-nitroso compounds instead of the maybe expected N-nitroso compound. I just wanted to highlight in a little bit more detail what I mean when I say a chemical class is covered and a nitrosamine will be predicted. So the nitrosamine rules will generate nitrization degradation products if they match a fragment of the compound or the API that's been submitted to Zenith and the conducive conditions match. So just as a reminder of the risk factors that I mentioned earlier. So for example, if I submit a gramine to Zenith and said, I have a source of nitrous acid and there's water present at an acidic pH, I would get this predicted pathway, whereby the amine undergoes acid catalyzed hydrolysis and to form DMA. This can then be nitrosated to give NDMA. There is also a direct pathway to NDMA via the tertiary amine. This pathway is also covered by one of Zenith's rules, but wouldn't be predicted as likely due to the tertiary amine nature. So I guess one thing that I just want to highlight at this point, because I sort of explicitly said it on the slide, is that we are aware that nitrous acid itself isn't necessarily electrophilic enough to react with the amine functional group. But because Zenith is a program, from a programmatical perspective, we have to define a structure for Zenith to match upon. And so we've chosen nitrous acid to represent our nitrosating agent aspect. And in reality, nitrous acid can form various active nitrosating agents, such as nitrosonium ion, depending on the pH, dinitrogen trioxide, protonated nitrous acid. So just to kind of highlight, we are aware that it's not always nitrous acid, but Zenith needs a defined structure. So that's what we've chosen. And as such, I guess my sort of conclusion for this bit is that Zenith can be used to assess the theoretical potential of your API to form a nitrosamine. Moving on to NDSRIs, detection of API-related nitrosamines, or so-called NDSRIs, as I mentioned earlier, added a further layer of complexity to the landscape for industry and regulators. It's no longer just formation of things like NDMA that are a cause for concern. And this all started with the detection of nitrosofuranoclin in 2021, and that led to the recall of furanoclin. This was followed by the recalls of propranolol, quinapril, and ephedrine, forgive my pronunciation. And these representative examples of NDSRIs that I've just sort of pulled together for this webinar, they've all been detected in a degradation setting, and they are all predicted by Zenith. So Zenith can also be used to assess the theoretical potential of your API to form an NDSRI. Now, I've been a little bit cheeky here, including irbesartan on this slide, and I've highlighted it in blue to try and denote that it's a little bit separate. This, of course, isn't technically an NDSRI. You've formed an oxime in preference to the API-related nitrosamine, but as I go on to discuss mitigation strategies later, I thought it kind of worth including, because sometimes it is important to know what might form preferentially, as opposed to just nitrosamines getting formed. In addition to the overall degradation products being predicted, Zenith will also go into all of the mechanistic steps for a given degradation pathway. For example, for doxylamine, which contains a tertiary amine, the mechanistic steps leading to the dealkylation to a secondary amine are all predicted, and the species is then nitrosated to, the NDSRI. For C-nitrization of amidines, there's an initial tautomerization. This species is then attacked by a nitrosating agent at the nucleophilic carbon, and the resulting C-nitroso compound then undergoes tautomerization, to the more stable oxime. And as I mentioned, that preference of formation of the oxime over formation of nitroso-irbesartan, so nitrization on one of the tetrazole nitrogens, is evidenced in the literature. So being able to assess the potential for nitrosamine formation of your compound and or whether it can form an NDSRI, this is useful knowledge, but what then? What if there is a nitrosamine risk? What do we do? What can be done to mitigate? Well, it's been estimated by Schlingerman et al. In a paper discussing the landscape of nitrosamines in pharmaceuticals that 40% of the APIs that they analysed and 30% of the API impurities that they analysed are potential nitrosamine precursors. Now, the amine functional group cannot simply be replaced or designed out of most APIs. This isn't a possible or realistic mitigation strategy for most examples. We can't just get rid of amine functional groups because of the unique and beneficial properties that the amines sort of display for drug development. API impurities are also not always easily purged or removed, or even, their causes detected from all areas of the synthetic route and manufacturing process. So what can be done? Well, excuse me. Regulators agree that cooperation between drug product manufacturers and excipient suppliers over the nitrite content of excipients can be a good mitigation strategy. Going back to this figure, which I showed, I've shown in a couple of slides actually, but I showed at the start, if we can remove or diminish one of the risk factors needed for nitrosamine formation, then the potential for these species is either much less or maybe removed entirely. where can we access nitrite levels in excipients? Well, one such source is the nitrite database hosted by LASA, who act as the honest broker in this project, and we facilitate the data sharing within a consortium of all these members outlined on the slide. At LASA, we don't generate any data ourselves, so we don't pose a competitive edge, and there's no sort of IP risk. And what this consortium aims to do is generate a comprehensive database for the levels of nitrites in a broad range of excipients to aid nitrosamine risk assessments. The consortium was established in 2020, and it currently contains nitrite levels for 105 excipients, the majority of those being commonly used in solid oral dosage forms, formulations of solid oral dosage forms. And there are 1,349 results. So that is to say that one excipient may have tens of nitrite results generated by several member organisations. And the consortium continue to add validated nitrite data to this database to ensure that it grows and it can be a trusted source of data. And the sharing of this data means that mean and max values for nitrite levels of commonly used excipients can be calculated, and there's been a recent publication on this in J-Pharmacide by Boetzel et al. How can this type of knowledge and data be utilised? How is this useful for us? Well, one of the aims of the database is to aid compiling an assessment of the risk of nitrosamine formation in drug products. If we were to consider the following tablet formulation, and this is purely made up, the numbers are purely made up just for an illustrative example, so please don't focus on the numbers, more the process that I'm going to go through. So if we had this formulation with these components and the associated composition of each part, the mean nitrite level, which you could either obtain experimentally or from the nitrites database if you were a consortium member, could be used to calculate the nitrite contribution for each component. This can then be totaled to give you the nitrite contribution of the excipients in your formulation. The following equation from the Boetzel publication can then be used to calculate the potential amount of nitrosamine that could form. Using the example formulation from the previous slide, so the number from the previous slide, if I was to assume that the API was varenicline, and therefore the potential nitrosamine was nitrosovarenicline with a molecular weight of 240 grams per mole, the EMA FDA proposed an acceptable limit. Well, up until last week, it was 37 nanograms per day. It's now been updated to 400 nanograms per day. I nearly missed that update. And if we followed the recommended dosing, that would mean a safe limit of 200 ppms. So if we plugged the relevant numbers into the equation, this would allow us to calculate the estimated amount of nitrosamines formed per gram of formulation. And it's probably worth saying that this would be a conservative estimate. And in this illustrative example, we would get 3.65 ppm, well below the 200 ppm safe limit. So these types of assessments can then be used to help support and direct the next steps and the necessary action that might be needed to be taken. And the ideas in the Boetzel publication, they've been utilised by Berardi et al. In their recent paper in Pharmaceutics. They looked at various case studies to ascertain how knowledge of nitrite levels and the subsequent amount of potential nitrosamine that can be formed can be used as a mitigation strategy. And one of their case studies demonstrated that change in the supplier of a single excipient, microcrystalline cellulose, so the green bar, the dark green bar, by changing that excipient to a lower nitrite containing version, could reduce nitrosamine formation by up to 59%, resulting in a nitrosamine formation level well below the acceptable daily intake of 178 nanograms in that particular case study. DFE Pharma, who are the authors of this work, they're an excipient supplier, and on their website they state that nitrite levels in our excipients are among the lowest in industry. Well, I can neither confirm nor deny that statement, but what could be foreseen is that these, no doubt, probably more expensive, low-level nitrite containing excipients could be used in high-risk formulations as a mitigation strategy. And the FDA supports these kinds of efforts by stating that supplier qualification, so a change of excipient supplier, and formulation design, so a change of the excipients used, to those showing a lower risk, are key mitigation strategies to reduce the risk of nitrosamine formation in a drug product. My final slide in this section just aims to kind of link those ideas around mitigating risk through knowledge of nitrite levels with knowledge from an in silico prediction. So in addition to the theoretical prediction of nitrosamines forming a degradation pathway, Zenith can also assess potential API excipient interactions or drug substance excipient interactions, allowing more of a risk-based stability assessment to be done, i.e., is this excipient suitable in my formulation? And this is because Zenith contains a database of around 350 excipient structures, and their associated degradants and impurities. So, for example, if I was to take varenicline again, and I wanted to formulate, excuse me, if I wanted to formulate that with acesulfame potassium, Zenith would tell me that this excipient has a nitrite impurity associated with it, which we would represent as nitrous acid for programmatical reasons, as I sort of explained earlier. And it would tell me, it would predict that nitroso-varenicline could be generated. The nitrite impurity has been added to excipients in Zenith in line with knowledge from the nitrites database, purely in a sort of a qualitative way, present or absent. But you could then use quantitative nitrite results, either experimentally or from the nitrites database, to then calculate the potential amount of nitrosamine in the formulation using the equation that I showed on the previous slide. And in this way, power of Zenith and the power of that nitrites database could be used to inform and support potential mitigation strategies. I'd like to wrap up this webinar with 4 concluding statements. So nitrosamine formation remains a challenge to assess and mitigate for all organisations involved in the drug development process, including regulators. And I don't think that's necessarily going away anytime soon. So we need systems to help us with that. And Zenith is one such tool that can assess the theoretical potential of an API to form a nitrosamine or an NDSRI via a degradation pathway. The nitrites database can then be used to help understand the impact and potential amount of nitrosamine formation in your formulation based on the nitrite levels in excipients. And together, those 2 systems can help inform and support mitigation strategies. So I'd just like to say a huge thank you to my very talented colleague, Grace Cox, who is the project lead for the Vitic nitrites database. And Grace helped me put together some of the slides for this final section of the webinar. I'd also like to thank the organisations that are involved in the Nitrites Consortium, because their collaboration and their sharing has really helped increase our understanding of excipients nitrosating risk. I'd also like to thank all my colleagues, past and present. Well, for the knowledge base, really, it takes a lot of dedication to build to build that huge library, and in particular for the recent nitrosamine section that we've added. Finally, I've listed all the references that I've used throughout these slides. To echo a sentiment that Maria said in her webinar a few weeks ago, thank you to all the authors who are publishing at a pretty phenomenal rate and helping to build and shape this landscape around nitrosamines. There's no shortage of information to cite, and in fact, the issue is trying to remain current because changes happen too quickly. And with that, I'll just thank you all for your kind attention and attendance at today's webinar. And I'll hand back over to Ken for a Q&A session.
Yes, thank you. Please continue to ask questions, and I'm going to again select a few to challenge Rachel with, and then she will get questions offline and respond individually later with that. So I'm picking a few of them. So one of the questions was, we focused in your risk assessment on entirely on nitrites. What about nitrates? Are nitrates a risk factor, or really don't have to worry about nitrates?
I don't know if I want to make such a resounding conclusion as you don't have to worry about nitrates, but I think there's
the answer could be you don't know, Rachel.
Well, I kind of don't know. I mean, the literature doesn't support that, you know, that there's not the literature on nitrates that there is on nitrites. So that kind of tells me something. I don't think that the formation of nitrates is as prevalent as nitrites. So that, I guess that is kind of my prevalent within formulations, I mean. So
well, there are a lot of nitrates around. The question is, can nitrates undergo the chemical degradation reactions? Probably not directly. So
not
then
directly, and not
can they, are there chains of reactions that might lead there? As you said, I agree with you, but thank you. That's good.
Yeah.
All right. The next question. This is less science and more regulatory.
Oh.
If Zenith decides that there really is a low risk, would a health authority say, you're okay, then you don't have to worry about it, or it's just an indicator?
Exactly. Zenith is a supporting tool. It is not a replacement for experimental evidence, and it is there to support conclusions and provide mechanistic rationale and just further evidence to sort of what I would call a full and rounded submission. I wouldn't like a regulator to take a Zenith prediction and say, oh yeah, everything's fine, because the theoretical space is a potential for everything. It doesn't mean that is what you will see in reality. Like any tool, Zenith has its scope, and you have to take the information from it in the correct context. So
yeah, and I think
would be my answer.
Nature of the question, I think the nature of the question is much like in toxicity things. We do a certain amount of risk assessment, and if a risk can be determined to be low enough, we can make an argument that it isn't a problem.
A weight of evidence
been able to use your database and types of calculations to do that. Okay. Is that something that are you aware of?
Oh, is that something I'm aware that people do? So yes, as a type of weight of evidence approach, yeah, people do use Zenith predictions to support regulatory submissions, but not as a they don't directly submit a Zenith prediction. Yeah,
right. And I think that's a fair thing. When you're making a case that something isn't a problem, one factor is to bring in some of this kind of scientific knowledge base, and this is a very good knowledge base. So it's not that it can't be used in regulatory, but like anything, it's only one of many factors.
Yes, okay. not on its own as a weight of evidence approach.
Is there a possibility, I always like the word possibility, of forming nitrosamines from tertiary amines?
Yes, I think there is a possibility. I think that there is enough evidence in the literature to show that there are, that there are mechanistic pathways and conditions conducive enough to see nitrosamines forming from tertiary amines. I don't think it's as likely. I think it's something like the rate of reaction is at least, is it 1,000 or 10,000? I can't remember the factor now, but it's a lot slower than secondary amines. But yes, I think
they're quite hindered for reactivity, and some of the
exactly
pathways are
it's
blocked.
possible, but maybe not probable is what I would say.
That I think is fair. All right, there were a couple questions. I hadn't thought about this one, but interesting. We've focused on this whole seminar series and the toxic things within nitrosamines. I suppose you can, in some cases, form carbon-based nitroso addicts.
Yeah.
Do they form, and then are they a toxicity risk when they form?
Yeah, so as I sort of outlined in one of my slides, yeah, there are examples of NC bond formation where you get nitrization at a nucleophilic carbon, rather than, even in preference to a potential nucleophilic nitrogen site, like I showed irbesartan as kind of an example of that. Toxicity wise, yeah, that's not my arena, I'm afraid. That's a direct nexus prediction right there. I don't know about the toxicity of like the oxime of irbesartan, actually.
Yeah, that's what I'm wondering about, whether that actually is considered a safety risk. Maybe we shouldn't ask too loudly, because all of a sudden we'll down parts per billion regulatory. So another question. Okay, so there's a question that's fairly detailed, so I'm going to read it out. For in silico tools, are the electronics, so I mean, a lot of things you're talking about are database driven rather than any specific calculation of electronic structure, but the question, I guess, is, are the electronics of the amines and nitrosating agents accounted for? Or is it solely on the presence of functionality? In essence, are known red flag exceptions brought in? What I guess it's kind of how much of this is database driven, and how much of it is rules, and how much of it is electronic calculations.
That's actually a good question. It's mostly database driven. It's mostly what is sort of in the literature, what structure activity relationships we can derive. So yeah, that is the main factor kind of driving the formation of those rules. Electronics, specifically for the nitrosamine section of the knowledge base, I would say that we don't take into account electronics at this moment in time, mostly because it's been such a developing area, it's been so fast paced that it's kind of hard to keep up. And yeah, it's hard to know kind of what to focus on. We also don't have, just to pick up specifically, and there was a point about, are there red flags for any, I can't remember what the phrasing was, but sort of specific compounds that we might include or exclude. No, we don't have sort of exceptions of that nature at the moment. What I will say is that we are accounting for electronics and properties much more in Zenith. So we have a BDE calculator, we have a pK calculator, and we are trying to utilize sort of those aspects of compounds more, not just structural matching. But for the nitrosamines, I think my sort of summary is, no, it is a data-driven exercise at this moment in time.
Okay, another question. So there's been a number of companies now that are employing antioxidant strategies, whether it makes sense or not. Is that something that Zenith can assess at all in terms of likely benefit or not?
No.
Not yet.
In a nutshell, no. We can't account for. Zenith is very much a present or absence type system, and it is a structural match. So Zenith doesn't handle quantities or concentrations. So it doesn't understand if you say, well, I've added 10% antioxidant, so what happens now? Zenith will just say, well, I'm just going to break that compound up into fragments and I'm going to tell you what it matches against. I'm making Zenith sound oversimplified, but in a nutshell, that's what happens. So no, it can't really account for sort of additional variables beyond structural, really.
Okay, another question a lot is focused was on drug-related formation of nitrosamines, but of course, there's also secondary reactions giving off low molecular weight nitrosamines. Is that something, the secondary type processes and tertiary processes that occur, is that something also calculated?
I'm not 100% sure if I understand the question.
So let's break it down to two things. Do you calculate the likelihood of formation of low molecular weight nitrosamines?
Yes.
So rather than just drug related?
Yes. Yeah. So you can form, Zenith will predict NDMA formation, for example. Yes, you can form the small dialkyl nitrosamines. Second
then with that was the some N-nitrosamines that are formed degrade themselves, and their levels will go down over time. Is the secondary degradation processes that are involved also calculated?
Yes. So Zenith, you can set Zenith up to predict degradants of your degradants, which is, I think, really what's being asked. Just to pick up on the levels, so amounts, no. Quantities is not something that Zenith can handle because it's a structural based match. But yes, successive generations of degradants, Zenith can predict, yeah.
And I think we'll be hearing in other talks that sometimes that if something can undergo degradation, it'll form a steady state level. And if that steady state level is beneath the specification limit or toxicity limits, then there may not be a stability problem, even though you're forming them. Okay, another question was when doing risk assessments with respect to levels of nitrite impurities and excipients, do you typically do average levels, or are you using worst case assumptions?
So that is a choice to be made. What I have usually seen in the calculations that I've seen in papers and things that I've seen in-house at LASA, we've been using the average, and we still find that is really quite conservative. But it depends how worried you are. I guess you could use the worst case, and then you can be really sure. But yeah, we typically are using average levels. But yeah. I don't have any specific reasoning for that, but that's what we're doing.
Well, it's also going to be interesting over time when you track these things that some of the excipient makers are adapting their manufacturing processes to use, let's say, water that is no longer has low-level nitrite impurities. And it'll be interesting to see if the level of nitrites gradually reduces. So hopefully the industry as a whole shifts that way. It'll make things easier for drug companies. All right, I think we're pretty much out of time. So I want to thank Rachel again for wonderful talk, and people can get access to her slides and this presentation through FreeThink's website or directly through LASA's website. And appreciate it. And everybody keep in mind, in 2 more weeks, we have our next talk, and hope to see everybody again. Thank you, everybody. Bye.
Thank you. Bye. See you, Ken.
Bye-bye.